Okay, let's get started. I know people are still jumping in. Okay, welcome everyone. I'm your moderator and one of today's presenters, Taichi Suzuki. I'm assistant professor at ASU's College of Health Solutions and Biodesign Center for Health Through Microbiomes. So my group studies the ecology and evolution of gut microbiome using humans and wild rodents. So I'd like to thank you all for joining Health Talks webinar brought to you by College of Health Solutions at ASU. Our college works to address the challenges facing people and communities to stay healthy, improve their health, and manage chronic disease. And these Health Talks are one way we serve the community with timely and relevant educational information that also provides continuing education credit at no cost. So before we begin, we have a few quick housekeeping items. So first, this session is being recorded and will be posted, and this will be posted on our website, asuhealthtalks.com. And after the panelist presentations, I'll be moderate a Q&A session, and we'll have about 20 minutes for Q&A. And please submit your questions using Zoom's Q&A feature and not the chat box. And regardless of whether or not you have requested CEUs, please register your attendance using the self-check-in link that's posted in the chat. So we have an accurate record of who attended today's webinar. And finally, you will receive a brief survey asking for your input about today's webinar. If you are requesting continuing education credits, this must be completed in order to receive credit. If you did not apply for CEUs, we would still appreciate your feedback to improve our series. So I'm very excited to introduce today's other speaker, Dr. Rosa Krajmalnik-Brown. She goes by Dr. Rosie, so I'll call her Dr. Rosie. So she's the director of the Biodesign Center for Health Through Microbiomes and a professor at the School of Sustainable Engineering and the Built Environment at ASU. But she specializes in the human intestinal microbiology and its relationship to metabolism and autism. So Dr. Rosie is a pioneer in research on gut microbiome and autism, and author of 11 US granted patents and more than 140 peer-reviewed publications. So she's also a co-owner and founder of 2 startup companies. So we are very fortunate to have Dr. Rosie with us today as we explore the impact of the gut microbiome on overall health. So the gut microbiome's role in health has become clearer over the past few years, yet there's still much to learn about it. And join our health experts for a basic overview of the gut microbiome and hear about ongoing research into what researchers have learned about its impact on health across the human body, how the microbiome affects diseases, and how to keep our gut microbiome healthy. You'll also learn about possible interventions to change the microbiome and recent clinical trials with humans showing how changing the microbiome improved symptoms of autism spectrum disorder. So with our 1-hour no-cost webinar, you'll get the information you need to understand the importance of our healthy gut microbiome. All right, I see this one chat. I think it's good. Okay, so I'll change the moderator hat to the presenter mode. So let's share my screen. Okay, do you see the right screen? Good, good. Okay, so I'll try to be brief so we have enough time for Q&A, but I'll try to give you the overview of microbiome research and how the microbiome affects health. I have no conflicts of interest. So as you may know, our body is covered with microbes. And when I say microbes, this includes bacteria, archaea, eukaryotes like protists and fungi, and even viruses. So collectively, these microbial communities, we call these microbiome. So let's start with some quiz. So if the chat is open, you can try to guess these questions. I'll give you a hint. So how many microbes live in our body? So the hint is our body, the human cells, it's about 30 to 40 trillion cells. What do you think the number of microbial cells that live in our body? Maybe sake of the time, and also it's hard to see the chat from my end, so you can think on your own and see if it's less, more. And the current estimate is actually 1 to 1. So this is pretty surprising, right? Our bodies are composed of half of our bodies composed of microbial cells. So how about our microbial genes? How many microbial genes are in our body? Is it less than? So hint is in the human genome, there's about 20,000 genes. Is this more of this, less than this? Is it about the same? Yeah, I can see the chats increasing. So the current estimate is about 150 times more genes. So why is that, right? Because for human cells, every cell has the same DNA, but for microbial cells, different species and groups carry different genes, and that is why there's more than 100 times more genes. So how heavy is our microbiome? And there's different estimates for dry and wet weight, but it's about the weight of our brain. So the point I want to make here is that there's many microbes that's living on our body, and at the gene level, we're 1% human and 99% microbe. So what are these microbes? What are they doing? And how are they affecting our health? And this question about how the microbiome affects our health is not new. We've been studying for a long time. And the classic approach is to grow them on these media and study them under the microscope. And if you see my mouse here, so if you want to see how many microbes are here, we have to use the shape of the bacteria or a shape of the colony or the color, right? But you can imagine there's a limit to this approach because two different groups of bacteria can have identical shape, and there's many unculturable microbes on our body. So what really changed in the past 20 years is this advancement in DNA sequencing technology that now we can characterize the entire microbial composition, including these unculturable microbes, through these sequencing methods. And the idea is pretty simple. So what we do is we take these microbial samples, break open the cell, extract the DNA, and we run through the sequencing machine. And at the end, we get this computer file of ATGC, of DNA nucleotides. And how it works is that different groups of bacteria can have a unique gene, okay, a unique sequence. So by counting the number of unique sequence, we can count the number of species or groups. By counting number of copies of these unique genes, we can calculate the abundance of the bacteria. So this approach really revolutionized the microbiome research. And there are a lot of global efforts to characterize the microbiome. So for example, the Human Microbiome Project in 2007, the aim is to characterize the microbial composition across different body sites. So from skin to oral cavity to the gut. And the Earth Microbiome Project is not only animals, but also characterize the plants and soil microbiome. The American Gut Project initially focused on the Americans, but expanded later to worldwide participants. So together with these global initiatives and individual labs, we as a field really discovered this underappreciated role of microbiome in human human biology or animal biology. So don't worry about the details here. I'll just walk you through the major link between microbiome and these traits. So for example, microbiome seems required for a normal development of different organs, blood vessels, to even bone development. So these work are mostly done, is based on germ-free mouse experiments. So these animals are born and raised in a sterile condition. And also another important role of microbiome is their effect on immunity. So there's two major ways. So one is a more direct way. So for example, like a biofilms, these are layers of microbes that physically being a barrier and protecting us from pathogens. And another way is microbiome can train our immunity. So it seems that it's important for early stages in life, being exposed to a variety of microbes seems to be important to have a normal immune development. And also microbiome is involved in circulatory system. So some microbiome metabolites can affect the cholesterol metabolism and can promote these plaques in the blood vessels that can lead to cardiovascular disease. And also there's some metabolites that directly affect the blood pressure regulation in animals. And also microbiome is important for digestion. So a good example can be like plant fiber digestion, right? Our human genome cannot break down fibers, but microbes can. And last but not least, microbiome is also linked to behavior. And Dr. Rosie will be touching on this later, but there's independent labs and studies that showed how microbiome can affect behavior like mood or anxiety type behavior. So if the microbiome affects this many aspects of our biology, maybe you're not surprised that there's many, many diseases that associated with the microbiome. And I'm not going to read through this list, but the good news, the flip side of this, is that it's modifiable. So compared to the human genome, it's easier to modify the microbiome to have a better symptoms and potential treatments for these diseases. So let me just introduce one study on obesity, this link between obesity and microbiome. So this is one of the classic studies for this DNA sequence-based microbiome. And these were led by Jeff Gordon's lab at Washington University. So if you look at the microbiome of obese individual and a lean individual, this is true in humans and mice, the obese individuals are characterized by this greater proportion of firmicutes. This is a group of bacteria in blue, and less Bacteroidetes. But just by this observation, we don't know the causality, right? Because microbiome can somehow promote obesity, but the other way is that it can be a consequence. So say obese individuals maybe have a greater food intake, maybe that's the consequence of seeing these compositional differences. So what they did is a fecal transplant experiment. So take the microbiome from the obese individual, do a fecal transplant to a lean individual, and they showed how lean individual can accumulate more fat due to this greater extraction of the energy from the diet. So when I first read this paper, it was pretty striking. So I plotted these proportion of these two bacterial groups on a map, extracting these healthy individuals from the published studies. And there's 2 points I want to make. So one is that all these composition, these are from healthy individuals that are not obese, okay? So that suggests that these healthy microbiomes can differ in different populations. And also as an evolutionary biologist, one way to explain this pattern is the need of energy extraction or the efficiency of energy extraction. So getting more energy from the diet is not necessarily a bad thing when the resource is limited, and especially in a colder environment in the past, right, compared to a tropical area. But that's only one out of the many explanations. This can be driven by diet, culture, genetics, and so on. So this leads to one of the fundamental questions in microbial research is to identify these determinants of the microbiome. And we know many factors affect the microbial composition and very complex. So we know there's a sex effect, medication like antibiotics, health conditions, and host genetics, which my lab specializes on, and environment, physical activity, and mode of delivery is an interesting one I'll touch later. But infants born with C-section versus vaginal birth, and they do develop different microbiomes later in life, and they're associated with the risk of asthma and allergies for the C-section on infants. So it seems, some research speculate that this can be due to the transmission of microbes from mother to offspring. And also there's a large effect of diet on the microbiome. So let me give you one example of the diet in microbiome. So this is a paper from 2014. So researchers gave 2 diets to participants. So one group received this plant-based diet, which is just vegetables, fruits. And another group received animal-based diet, like meat, eggs, and cheese. So you can see these fiber intake differs between the 2 groups. So now if you look at the microbiome changes, plant-based diet didn't affect the microbiome that much. But animal-based diet, you can see this changes in microbiome, and it's pretty rapid. And this seems true for all the follow-up studies that diet have a fast response, But when you go back to your original diet, it comes back to your original composition. So it's also stable across time. So diet is a one intervention that we can change the microbiome composition. And another important topic in microbiome research is this: where did our microbiome come from? So this is a question about the transmission of the microbiome. So all mammals, when we are born mostly sterile, and we first get our microbes from mother's vaginal canal and then mother's skin. And the breast milk will help recruit the beneficial microbes. And as we develop, we get microbes from other individuals in the environment. So here's a recent study showing these transfer events of microbes. So I'll walk you through this. So this first column, the same individual. So this is showing a microbial samples collected from the same individual across different time points. So you can see majority of the strains, you can detect the same strain at different time points. So this may not be surprising, right? These microbes can be stable. And this mother offspring column, it's showing there's identical microbial cells you can identify between mother and offspring. So this is suggestive of vertical transmission. But we also see transmission within households, within village, and sometimes even between populations. So this is this study is looking the transmission for one generation, but my group studies this transmission across many, many generations. And we found that some microbial species may have transmitted through thousands of generations since out of Africa migration pattern. So here's a visualization of human genetic relatedness from the 6 countries. So basically, this is just showing, for example, individuals within Africa are more genetically similar to each other than to individuals from Europe or Asia. So this is not surprising, right? But when you look at the gut microbial species, you can find some that look very similar to the human genetic relatedness. So this suggests that these microbes may be following our migration by transmitting among the relatives. But in contrast, there are microbes that show no patterns, and this is suggestive of panmictic species that they've been transmitting between species. So my group is interested in what's so special about these vertically transmitted microbes and how they affect the health. So just to summarize what I told you today, is that these advancements in DNA sequencing technology have revolutionized the microbiome research, and the gut microbiome can affect the normal development and overall health. And disentangling these different factors that affect the microbiome is very important, right, for controlling the microbiome. And we know that we acquire these microbes from parents and also from people around you. So one take-home message might be that there's no single healthy microbiome, that healthy microbiome differs by population and by individual. So the gut microbiome is a potential target for more personalized medicine and disease management. So for that, thanks for listening, and we'll hear more about the intervention aspects of the microbiome research from Dr. Rosie next. Okay, so let me stop sharing. Okay, so as we set up, in the moment we'll hear from our next presenter. But first I want to remind you that if you haven't already done so, please register your attendance using the self-check-in link posted in the chat, so we have an accurate record of who attended today's webinar. So now we'll hear from Dr. Rosie.
Thank you, Taichi. And I'm going to keep the same title because I'm just continuing the talk and the conversation that Taichi so nicely started. It is my pleasure to be here. Hold on. And I can't advance my slides. Okay, now I can, sorry. Conflicts of interest. I have several patents on MTT or FMT on autism and autism diagnostics. I'm the co-founder and co-owner of 2 autism-related startup companies. And I've received some funding from Finch Therapeutics and from GBAT, which is gut-brain axis therapeutics. And so as Taichi so nicely introduced us into the gut microbiome, I just wanted to mention a few things of how the gut microbiome helps to our health. And some of the things that the gut microbiome does to help us is the gut microbiome microbiome helps us digest our food, which Taichi mentioned. It produces vitamins for us. It protects our intestine. It protects us from attacks from pathogens. It produces short-chain fatty acids and some important metabolites that can interact with other parts of our body. So for example, the gut microbiome through these chemical signals interacts also with the immune system, with the brain through satiety and a few other signals, and it also interacts with the liver. And one of the things that my research team has done for many years are interventions of the microbiome to understand how some of these things work. We don't work with mice, we work with humans, which are a lot more important than mice, in my opinion. And one of the ways that we approach understanding and learning more about how the microbiome works or contributions of the microbiome is by making changes in a clinical trial and in longitudinal designs. We are the first team that published on how the microbiome changes after bariatric surgery. And we also have done work on diet and how the microbiome changes with diet and probiotics and prebiotics. And the one intervention that I don't have listed in here are antibiotics, which is also an important one. But today I'm going to talk to you about treatment that we devised that it's called microbiota transplant therapy that we've used mainly for autism. So autism, or ASD, which is how I mention it in a lot of my slides, it has a huge societal impact. The diagnosis of autism has been increasing dramatically in the United States, which you can first see on this graph. And also to give some context, in the year 2000, the diagnosis by the CDC was 1 in 150 school-age children. And the diagnosis in 2023 was 1 in 36 school-age children. The diagnosis for autism is based on behavior. There's no biochemical test for diagnosis, no biochemical marker, and there's no cure or treatment available for the core symptoms of autism either. The estimated cost in the United States is about $461 billion per year. And so Taichi really nicely mentioned some of the factors that affect with the microbiome. And in this slide, what I have is some of the factors that have been pointed at that could be causative of ASD. And we know that genetics is one of them, but only a small percentage of individuals with ASD fall in this category. Environmental exposures is another one. And one that my lab has been investigating for the past about 15 years is the microbiome. And I do want to point out that, as was mentioned before, the microbiome interacts with these other two components. The microbiome interacts with the host, with the genetics. The microbiome interacts with the environmental exposures. So the microbiome, besides being something that might be contributing to this disorder, is also a signal of the other two components that are possibly causative also of the disorder. I was convinced that investigating if the microbiome had certain interaction with the disorder with ASD as a junior faculty, as an assistant professor, by two different points of evidence. And one of them was a study published by my collaborator, Jim Adams, where she showed that gastrointestinal symptoms in autism correlates with autism severity. So what you're looking at in this graph is on the y-axis is a score of gastrointestinal severity. The higher the number, the more intense the gastrointestinal problems are. And on the x-axis, you're seeing autism treatment evaluation checklist. Also, the higher the number, the more severe the diagnosis is. And you can see that there's a correlation between these two. The other point of evidence that convinced me that this was a question worth investigating was a study that was published in 2000, where children with autism were given vancomycin, which is an antibiotic that it's not absorbed and makes it all the way to the gut. And you can see in this line their behavior at baseline. And during treatment with vancomycin, the behavior improved. The higher the number, the better this behavior is. And what happened with this treatment is that the kids stopped taking vancomycin, and there was a follow-up later where you can see that the behavior regressed after they stopped taking the vancomycin. But just the fact that the vancomycins had some effect on their behavior was a really point of evidence that this was a question worth pursuing. So many years later, now we know that the microbiome affects the gut and the brain health. And on the right, I'm going to show you first what we think about healthy gut function. When we have a healthy microbiome, normally we have lots of microbes. We call this high diversity. These microbes, as I mentioned before, will produce vitamins, will produce butyrate, which is a source of energy for the gut cells. And this will be mainly a balanced microbiota that will also help with normal tight junctions. And this microbiota interacts, like I mentioned before, with the whole body and with our nervous system. When this microbiota is altered, we normally have lower diversity, abnormal gut function, GI symptoms, inflammation, and production of a few metabolites that are not exactly beneficial. I'll talk about a couple during my talk. And this also possibly provides alterations in tight junctions and something that has been called in this area leaky gut. And this can lead to abnormal central nervous system, anxiety, irritability, and inflammation. So when we started looking at the microbiome, the very first thing we did was comparing how does the microbiome of kids with autism looks in compare to the microbiome of typically developing children. And we did this. Since then, there's been many studies. You will have many different findings because it's a very heterogeneous disease. But there's a few important things that have been repeated in all these different studies. And one of them is the increase of harmful, I put it in quotation, microbes like Stutterella and Clostridium. We actually had a different approach where we were looking to see if there was a lack of beneficial microbes, which is exactly what we found. So we found in our cohorts that these kids had lower diversity and they were missing microbes that are important for fermentation and production of these short-chain fatty acids like Bifidobacterium and Prevotella. And there was a study more recently, which is a multi-analysis of many of these different studies. It was a meta-analysis that put together all the data from these studies. And one of the things that they found was that there's about 590 microbes that are more commonly associated with children with ASD, and about 170 that are more commonly associated with the control counterparts. They also showed that some of the changes that I'm going to show you were repeated throughout this whole cohort study, meta study. And so because one of the things that we saw was that there was lower diversity in our children, we devised a treatment where one of the things that we wanted to do was add beneficial microbes to their guts. And so how did we do that? We devised a study that we call, in the paper, we called it microbiota transfer therapy. Now we call it transplant. It's a microbiota transplant therapy. We treated 18 kids. The treatment was, it started with 2 weeks of vancomycin, the same antibiotic that I showed you before. Then there was a cleansing step, a cleansing similar to what you do for a colonoscopy, which is Mobi Prep. And then we had a high dose of microbiota that it's a microbiota that is produced from fecal samples of very healthy individuals, but it's not feces, it's microbiota purified from fecal samples from very healthy individuals. And then we have a low dose for 8 weeks. And so we did this treatment, the whole treatment is 10 weeks. We collected a lot of stool swab, blood samples, and we also collect a lot of behavioral assessments and GI assessments. The treatment lasted 10 weeks, but we also did some assessments 8 weeks later to see if we saw some improvements with the treatment if the improvements remained, because we had seen on the vancomycin study that the improvements did not remain after the study. And then I'm going to show you the data, and you will see that besides doing the 8-week follow-up, we also did a 2-year follow-up. So the first highlight that I want to point out from this treatment, and I do want to mention that this was open label, so everyone was being treated and everyone knew that they were being treated. But the first highlight is that we had 0 dropouts. So if anyone in the audience has run clinical trials, you know that that's a huge achievement in itself. 0 dropouts, everyone finished the treatment. Now I'm going to show you some of the results of this treatment, which of course were very exciting. And so the first thing that I want to show you are the gut benefits after the treatment. And on this graph, what you're looking at is a GSRS assessment on the y-axis. The higher the number, the worse the GI symptoms. The lower the number, the best the GI symptoms are. And normal GI is a number 1. And so you can see that most of the children that participated in our study started with pretty severely GI symptoms. Here, after 10 weeks of treatment, a lot of the GI symptoms improved dramatically for most of the children. A little bit of this got lost after 8 weeks, but they're so much better than when they started the treatment. And on the 2-year follow-up, some of the improvements got lost, but they're still much better than in the beginning of the trial for most of the kids. Some of the things that improved: abdominal pain, diarrhea, and constipation. So that's GI, which was in itself very, very exciting. But when we look at our behavioral symptoms, this is CARS, which is Child Autism Rating Scale. And what you're looking at in here on the y-axis is how this scale is labeled. Beyond this, above this line, it's severe autism diagnosis. Between these 2 lines, it's mild to moderate, and under this line is minimal to no ASD diagnosis anymore. So you can see that the majority of our children started on the severe category. 10 weeks after treatment, many of them improved to mild to moderate, and some of them even to minimal to no, or on the line. 8 weeks, they were doing much better. And one of the reasons why we did the follow-up 2 years later was a few of the parents of these children were calling our study coordinators to tell them, my child is doing better and better and better. So Jim Adams, my collaborator, said, Rosie, we need to document this. And I am really glad we did it, because we can see that 2 years later, they were doing even better than at our follow-up treatment, which this was very exciting. You can see in this graph that some of these children actually are not even considered in the spectrum anymore. The media went pretty crazy with this. So we had a lot of press releases and a lot of excitement inside and outside of ASU. The Economist wrote about our work when this happened, which was a very exciting moment. Okay, so I show you now the behavior and the GI symptoms. Now let's look at what happened with the microbiome. So with the microbiome, in this graph, what I'm showing you is the similarity to the microbiota that we provided. Unweighted unifract analysis, it's a measurement of dissimilarity. On this green line, we have a comparison to average neurotypical children, which were age-matched. They were our controls, which were not treated, but we just had them as a comparison. And you can see here the children before they started the treatment, and you can kind of assume that the differences between one child and the other is about 0.5. The lower the number in this graph, the more similar things are to what we're comparing them. And here we're comparing them to their donor. So you can see here that after 3 weeks, these children were now more similar to their donor, which means that the microbiota that we're putting in is colonizing their gut. And even though this similarity gets lost a little bit after week 10 and week 8, they're still more similar to their donor than they are to the person sitting next to them. However, when we did the 2-year follow-up, they're not similar to the donor anymore. They're not similar to the donor anymore. They've developed a new microbiota, which I will show you now that it's different to the beginning microbiota, but this is a beneficial and healthy microbiota. I'll show you that in the next few slides. So what happens is that when we look at diversity, which is one of the things that we wanted to improve, because if you remember, that's one of the things that was lacking in our cohort. In this graph, faith diversity, it's a measurement of diversity. The higher the number, the better the diversity. The green line are our typically developing children. This line is our initial microbiota donor. This other line is our maintenance donor, which is the 8 weeks that was done throughout. And you can see here that the diversity improved slowly with the treatment. At 3 weeks, not so much because of the vancomycin. 10 weeks, the diversity is improving. At the 8-week follow-up, they're almost as diverse as the typically developing children. But at the 2-year follow-up, the diversity increased even more, and they have even higher diversity than our typically developing children. So this is one of the signals or signs of a healthy microbiota. And so what we think that happened is that we changed the environment with this treatment, and now these children had the potential of recruiting and retaining beneficial microbes in their gut. We also saw important changes for specific microbes. Bifidobacterium, which is a beneficial commensal microbe. We can see here our typically developing children, the green line again. And here is where our children started, and you can see increases at week 3, week 10, week 8, and at 2 years later. Prevotella, which is a microbe that we saw mainly depleted in our first study. It's mainly depleted in most of the children, even though it's kind of high in 3 of them. And it's obviously much slower than our typically developing children, increases at week 3, week 10. And even though it decreases a little bit at week 18 and 2 years, it's still much higher than our typically developing children. And the sulfovibrio is another microbe that it's a little bit controversial, but we're following it because it's involved in sulfur cycling, and we're interested in that. Also is a microbe that increase with treatment. So we saw great improvements in the microbiome composition. We saw great improvements in GI, and we saw great improvements in behavior. And so as good scientists, we wonder, what is the mechanism? What's happening? And so there are many hypotheses by which possibly the microbiota in the gut can be communicated with the central nervous system and with the brain. And the hypothesis that my team has been pursuing for a few years is chemicals, metabolites that are produced by the gut microbiota that can travel through the bloodstream and communicate with other parts of the body, including the brain. And so there's some microbial metabolites that are either precursors of neurotransmitters or that interact with neurotransmitter production. So this is one of the hypothesis that we've been pursuing. So luckily, we collected fecal samples and blood samples in our trial. So we looked at plasma samples, and what I'm showing you in this figure is a PICO-A analysis, and I'm going to guide you through it with me, so you can hopefully see what I'm trying to show you here, which is that what happens in the gut doesn't stay in the gut, and we were able to see changes in plasma of the metabolites. So in this figure, what you're looking at is on the circles in red is our kids at baseline, or kids that were going to be treated at baseline. On the squares here on red, sorry, on the squares in red are our typically developing children, which are what we're comparing with, or baseline. So you can see here our TDs are on this side of the graph, and at baseline we have here our children that are going to be treated. And you can see that after treatment at week 3, they're starting to migrate towards being more similar. Each dot is the metabolome. It's the whole composite of the metabolites that we're measuring in plasma. And so you can see that the metabolome becomes more similar to the typically developing children, and that the metabolome of these kids after 10 weeks completely migrated to this side of the graph, which is where we have the typically developing children. This is, of course, very amazing and exciting, because then we can see that there's systemic changes that happen with this microbiota transplant therapy. And so some of the metabolites that we saw that were different is paracresol sulfite. And 4-ethylphenyl is another metabolite that has been seen in ASD studies with mice. which are possible signals of unbalanced microbiota, and we know that interact with the dopamine production. So here's, a good signal of some of these microbial changes that might be happening that we can hopefully change with microbial transplant therapy. And I am not I'm not showing those data here because I want to have time to talk to you about something else, but we actually saw paracresol sulfate changing with the treatment also, which is important. It was high before treatment and it got the relative abundance came down after treatment. So we also did some more analysis on the microbes in the gut doing shotgun metagenomics. This was work done by Kem Nirmalkar, who is a very talented research scientist in my group. And we were able to better define some of the species that we found, which again, we found Prevotella and Bifidobacterium and the Sulfobibrio changing with the treatment. But we also found that there were genes that encode for folate production, genes that are involved in oxidation stress or oxygen detoxification that increased with treatment, which is a good thing. And we also saw some genes involved in glutamate production and genes that take part in sulfur metabolism that change also with treatment. So these are important hypotheses that we will be following in our studies. So I hope that with this, I convinced you that we had this treatment, that with this treatment, we changed the microbiome. And we changed the microbiome drastically to be more similar to the donor, but in the long term, each individual had a new personal beneficial microbiome. And I showed you also that these changes led to systemic metabolite changes that show that changes in the microbiome affect overall metabolism. We saw GI improvements that lasted for most, but not all participants. And we saw that autism symptoms generally improved over the next 2 years in most, but not all participants. Again, this was an open label trial, and for sure there's some placebo effect possible here. So what's next from this? It's, of course, a phase 2 trial, which is a trial that has a placebo arm, and we're doing that. And so right now we're running, we have already run 3 phase 2 studies, one with adults, one with children, and one with children with Pitt-Hopkins syndrome. And currently, we're running a children dosing study and a study with children and adults with Pitt-Hopkins. And so as a little highlight, because I know you probably want to hear a little bit about our phase 2 trials, our highlight is a little bit of data that I'm going to show you on our adult study. This was a randomized, double-blinded, placebo-controlled trial. We had 56 participants enrolled, 51 completed. So not perfect like our child study, but very high completion rate. It was safe. The treatment group had fewer or less adverse effects than the placebo. It was effective. We have ASD symptoms, medium effect size after 10 weeks, and large effect size after 18 weeks, and GI symptoms, medium effect size after 10 weeks. And so our CARS graph, I want to show you in this study, we did two arms, one arm that was initially on placebo, but later on we switched them to treatment, which is a great way to retain patients and make sure that everyone gets the benefit of the treatment. But on the second arm, when they were switched to treatment, they did not receive vancomycin. So in this graph, what you can see on the blue line are those that were treated initially, and you can see that there's huge improvements at the end of part 1, which is the end of treatment, and it's higher improvement than the placebo arm. And this group that was treated first with vancomycin and then with the microbiome continue improving at the end of part 2 because they were treated longer, actually. So this is one of the reasons we're doing a dosing study. And then our follow-up, the improvements continued just like we saw with our children. And 18 months later, improvements improved more too. On our placebo arm, this is when they were not being treated. This is just placebo. And then they were treated from end of part 1 to end of part 2. So here, there is treatment here, but without vancomycin. So you can see improvement, but without the vancomycin, it looks like this is less stable than what we saw with vancomycin. So A, it looks like this works also with adults, which is wonderful, and B, it looks like we do need vancomycin for our treatment. Another thing I want to show you from this treatment is that this treatment with adults was probably one of our most challenging things we've done because it happened during COVID. And besides the fact that it happened during COVID, we had to switch the source of our microbiota donors. So we have two different sources of microbiota. One of them is Finch Therapeutics right here. And then the other source of microbiota is University of Minnesota. In this figure, what you're looking at is a distance matrix where each dot that you're looking at, it's the overall microbiome of a patient. And the closer the dots, the more similar they are, and the farther apart they are, the less similar they are. So in this circle, you can see all our participants when they started the study. This is our baseline for everyone. And then they were treated with vancomycin, and you can see that when they were treated with vancomycin, they all migrated to the same area of the graph. So they're very similar when they're treated with vancomycin. After we treat them with vancomycin, the patients that received the human donors microbiota migrated to be closer to those human donors. So you can see that the microbiota is similar to their donors. And those that were treated with the Finch donors migrated to be more similar to the Finch donors. So you can see two things here that this is effective and we achieve similarity, and also that microbiota matters. And on the right side, you can see the placebo, and you can see there's no groups, there's no migration, there's no things that are similar other than here, or donors. These are the green donors and these are the black donors. And so I know I'm running out of time and I want to leave enough time for questions. So I want to thank, this is my research team, my amazing research team, Dr. Daewoo Kang, who did most of the phase 1 studies that I showed you before, and now is an assistant, almost associate professor in University of Toledo. Jim Adams, my best collaborator, Dr. Kamla Nirmal-Kar, who did a lot of the work that I showed today too. A lot of families, a lot of donors, a lot of collaborators, and a lot of research funding agencies, and all of you for listening and for being here still. And with this, I conclude my presentation, and I know that we have a moderator that will help with questions.
Thank you, Dr. Rosie. That was great. And I hope everyone enjoyed. We have a lot of good questions. So we will move on to the Q&A session. Yeah.
Do you want me to keep it, or do you want me to stop sharing, Taichi?
I think go ahead and turn that on.
Yeah.
What?
Stop.
Yeah, stop sharing.
Okay.
Thanks.
And when we need, like references to figures, we can go back.
Okay.
So we have a lot of great questions, and we'll do our best to answer them. So I'll go from the earlier questions. And this one, it might be for you, Rosie. I'm not an expert in cancer microbiome, but one question from Yasmine. So are there any recent studies that have been able to correlate disruption in the gut microbiome and GI-related cancers, such as gastroesophageal and colorectal cancer, especially because of the alarming surge and early onset of these cancers in the population?
I don't know, and I like to say I don't know when I don't know. I do know that the microbiome, there's been a lot of work with the microbiome and immune therapy for cancer, and the microbiome is really important for good outcomes of immune therapy for cancer that I know. I don't know if there's a correlation between colonic cancer and the microbiome. My guess is yes. That's an educated guess, but I'm not the expert on that topic. So,
no, thank you. I agree. My guess is also yes, because for correlational, it can be bidirectional, right? There's some microbiome dysbiosis, oxidative stress, more DNA damage, cancer, and other way around, right? Cancer, more cascade effect of microbiome.
It's true. And for the gut-brain connection, I showed it on my figures, but I don't think I mentioned that. It's a bidirectional connection. It's gut-brain and brain-gut. That's something to also keep in mind. So there's this collaboration, with the microbes. Like the microbes interact with the host, and the host interacts with the microbes. It's a collaboration. It's a two-way street. And that's also something to keep in mind. So when we see results from microbiome studies, we can't say that it's causative many times, especially on cross-sectional studies. It could be a signal we're detecting of something else happening with the host, you know? So the microbiome could be maybe causing something, but the microbiome can also be a signal of something else happening.
Exactly. Okay, moving on. Have there been studies to see if providing C-section babies with bolus of fluid they would have received had they been
Yes.
delivered vaginally would be beneficial?
Yes, yes, there have been studies. Dr. Dominguez Bello has done studies with that, where they actually swab the vagina of the mom and swab the babies with that. So at least she's one of the researchers that I know that has done that. I believe, if I remember correctly, Rob Knight's baby also was swabbed because his wife had to have a C-section, an emergency C-section. Rob Knight is a very famous researcher in the microbiome world.
Great. Next, are you saying if as a mom I maintain a healthy microbiome, it will impact my family?
Yes.
And yes.
Yes.
So it's called maternal effect. And
yeah, it also affects your family because what you need to do to have a good microbiome is eat well, right? So if you're eating well, that's an example, and that's something that you're hopefully getting all your family to do. So, you know, that's, yeah. No pressure, but yes. And also for the dads, by the way, this is not a comment just for the moms, for the dads too.
Exactly. Microbes transmit within the household, and there's studies, even dogs to humans, and this is also pets too. So Magdalena, great talk. My question is for Taichi. What's the microbe that is vertically transmitted that you find most interesting? What are the microbes that might have been lost with the Industrial Revolution, and what impact might that have in population health? This is a great question, and I guess one that's vertically transmitted, the most famous one is Bifidobacterium, and this genus or within some species are used as probiotics as well. And in the human breast milk, there's I'll be brief, but there are components that specifically made these complex sugar that's made for these bifidobacteria. So that's one of the only bacteria, or the only bacteria that can break down. So as a human body, we are providing to recruit certain bacteria. And so bifidobacterium is one that's vertically transmitted. That's interesting. The potential coevolution with humans. And for Industrial Revolution, so this is there's a lot of discussions on this, that if you compare rural and urban populations, the microbiome is different, and the diversity, the richness is lower in urban populations. And I think in general, a lot of fiber degrading microbes has been lost or low abundance, and that's mostly due to our modern diet. And is that a good thing, bad thing? Is pretty controversial. Some people think our microbiome might be adjusting to the modern processed food, and some people think losing these microbes can be impacting the health negatively. So this is active area of research. So next question, we'll do our best to go through. That's good. A lot of questions here. So from Scott, how might microplastics affect the microbiome? I'm thinking about microfilms forming on plastic humidifier parts and pet bowls. Yeah.
So one of my former PhD students actually recently did a study on microplastics in masks and how that affects the microbiome and how the microbiome interacts with the microplastics in the masks. So yeah, there's an interaction. It's a chemical. Any chemicals affect the microbiome. That's another thing that maybe Taichi and I didn't mention it, but chemicals in the environment and the drugs we consume also interact with our microbiome. So our microbiomes actually change the drugs and sometimes change the pharmacokinetic of the drugs. So it's the same with microplastics, that probably the microplastics interacts with the microbiome and the microbiome interacts with the microplastics. How? I don't know specifically, but there is probably an interaction.
For the study in autistic individuals and MTT, what was the median age of those enrolled in the study?
I don't know right now that I can tell you the median, but it was 7 to 17. We wanted to do it with younger children, but we had to get FDA approval to do it. As we had to get an IND, which is a new drug, investigational new drug permission. And so the FDA didn't allow us to go like that low in kids, which they will in the future because we've already provided safety. They said you have to start, if I remember correctly, it was 6 or 7 years old, because the younger the kids is high risk, high reward. And so, yeah, but it was like 7 to 17, if I remember correctly.
Right.
We're trying to recruit younger right now, about 3 years old to 17. And also we've had a lot of interest. So whenever we recruit for our studies, we get these questions too, of course.
Mm-hmm. So great talk. Are there any current studies on microbiome and autism in adults? Additionally, have there been studies examining differences in the microbiome across various ethnicities? So maybe the autism side, you can go first and I can follow up on this.
So we have this one in adults that we need to publish it. Like what I show you, it's not published yet, but we will publish it. We're working on that. We have a manuscript in preparation. And it's harder with adults. We actually were not sure that we were going to see a response, because the idea is that early intervention is actually what counts, and the microbiome is supposed to be more stable with adults. But we did this study with adults with autism, and like I said, we're hopefully going to publish it really soon. And that's, I don't know about others. Most of the people focus on children because it's in theory easier to change the microbiome in children. We did not see a lot of difference in our first study between the ages, which was also motivation for us to also work with the adults.
Mm-hmm. And microbiomes have been studied across different ethnicities. And it is challenging. There's not that many studies, but there are differences. But these differences can be due to diet, cultural aspect, and it's very difficult to control. And self-reported ethnicity may not reflect the genetic ancestry or that size. So it is mixed, or still a lot of work to be done. And our lab is also funded to do this research recently from the state. So we'll keep you updated. Okay, so what type of supplementation is suggested, let's say for anxiety? I
don't know. So I wish I knew. I don't know.
No, but that's the right answer. And I think from your work, a lot of those are fiber degraders too. So that's one thing we learned from microbiome research in general, is it seems like promoting these fiber degrading bacteria is one way, but yes, direct link, I agree. That's a good, honest answer. Which behavioral tests were used for the study?
There were mainly autism assessments. The reason I showed CARS, it's because CARS was done by an external evaluator, which is a child autism rating scale. But we also had SRS, ABC, and another assessment that I can't remember right now, which I think it's PGIR, which are parent global impressions. Some of these were done by the parents. They were done more often by the parents, like some of them almost every week. But we know that with the parents, sometimes there's some strong placebo component, which is why I normally like to show the CARS, because that's done by an external evaluator.
Mm-hmm. So we do have to end at 1 p.m. sharp, but we'll keep going. So Dr. Hart, how do you enroll participants?
Send me an email and I'll send you a link, and that link puts you actually in our mailing list. And we advertise the study and people apply, and that's our study coordinators randomly pick the participants. But right now we have our studies full, and it's been a blessing, because running clinical trials, recruiting is usually a challenge. But for us, our child study, we had 50 spots, 1,500 applications. So yeah, but we're hopefully getting close to running a phase 3 trial, which will have more participants. So if you're interested, send me an email, and I can send you a link where you can sign in and get notifications for the trials.
Maybe last one or two, Dr. Sklar. Since there are symptoms in children at an early age and their diet is limited, are there differences in toddlers compared to adults with the treatment?
There are differences between the children and adults for sure. And diet is a component that we're investigating also. That's a very good point. And we looked at the diets of the children in the beginning of the study. On our very first study, we didn't look at it later, but now we have a design where we look at the diet also after the treatment. And I am pretty convinced, I have a very strong hypothesis that once they feel better, they start having a more diverse diet. They're starting to explore more on different things to eat. And that also helps the microbiome.
Okay, I think that's all we have time for today. So please send us email. We can answer too. And if you have not already done so, please register the attendance using the self check-in link. And we encourage you to follow the CHS on social media so you can learn more about this type of opportunities. And our health talk series will continue on April 10th when we'll discussing exploring public health cases and impacts before the Supreme Court. And details and registration will be available through the asuhealthdocs.com. So thanks again for you all to joining today, and have a great rest of your day. Thank you.
Good job, Taichi.
Thank you. You too.
Bye.
Bye. Thank you all.